All right. We are being recorded. The lecture will be on YouTube after we finish. I hope you had a good weekend. Maybe some of you studied, some of you will still study a bit later for this chapter, chapter 2. So we have been talking about water and various properties of compounds which come from water environment. So I'm gonna share my screen. Again, I want to emphasize that we have these learning goals, right? We have these learning goals, and this is what we go by. This is what we go by. This is what I focus on in our discussions, and this is what I will test you on. So keep that in mind and always check with those. Always check with the first slide. Again, let's try to make this interactive. If you have any questions, always stop me and ask for clarification or any further information. All right, so we finished last time on Friday. We finished by chatting about hydrophobic effect. So remember, this is the effect which is responsible for making these hydrophobic molecules with hydrocarbon side chain aggregate to form one larger structures, and that comes from the decrease in the organization of the molecules of water and increase in less significant decrease in entropy. Now, when you have lots of these hydrophobic molecules, it's possible for them to aggregate into these what's known as micelles, right? When you have very high concentration of these amphipathic molecules. So I asked this question last time. You told me this is how soap works. Yes, exactly right. So soap, right, is the consists of fatty acid molecules which have polar head groups, which will be on the outside, and hydrocarbon chains will all be inside. And all the dirt and all these lipophilic molecules which you want to get rid of, take off your hands, all the viral particles, COVID, right? You want COVID to go inside there, into this micelle. Get it off your hands. So this is what happens with larger concentration of these lipophilic molecules. And drugs work the same way, right? So why do drugs bind to macromolecules? Why do drugs bind to enzymes and various receptors in the body? Well, how do hormones work, right? So they also bind to receptors in the body. And they take advantage of this hydrophobic effect, right? Same idea. So if you have an enzyme, so when we study the proteins, we will find out that amino acids which are on the outside of proteins or enzymes are primarily hydrophilic, right? They're designed to interact with water. And so water molecules will bind to them to a certain degree. And here's our drug, which is also surrounded by molecules of water. This is what's known as a hydration sphere. Hydration sphere. You can write this down, very important term. Hydration sphere. and this hydration sphere of an enzyme, right? And so when these come into contact, you allow to lose some of this hydration sphere and create these points of attachments, which come at the expense of displacement of these molecules of water, and all these molecules of water will result in the entropy going up, right? And that's what will make the drug bind to its biological receptor. All right. So just like water changes the properties of molecules dissolved in water, and the molecules themselves, once they get in the water, they change the properties of water. Now there's this colligative and non-colligative properties. So colligative, basically what that means is that they do not depend on the nature of the solute, just the concentration. And that's the boiling point, right? So for example, if you have a sodium chloride, table salt, if you drop this in water, or if you have potassium sulfate, drop this in water, as long as they're the same concentration, they will increase the boiling point of water by exactly the same amount, exactly the same degrees, sorry. Same with melting point, right? If you want your roads to stay unfrozen, right? So safe in the winter, it doesn't matter what salt you dump on it. What matters is how much, right? The concentration. And that there's also something called osmolarity. So we'll talk about this in a moment. Now, other properties such as viscosity, surface tension, obviously taste and color. So these are non-colligative. And so why is this important? So we'll talk about cytoplasms of the cells, right? So cytoplasm, you can treat cytoplasms of the cells as vessels, right? Something that it's a porous vessel that allows for salts in and out, water in and out. So it's very important. The concentrations of salts on the outside and inside is very important. And so the properties of water become very important as well. So let's remind ourselves about this osmotic pressure. This is from general chemistry, right? So what causes that is that water will move from the areas of high water concentration. So in other words, if you have a dilute salt, right? Dilute salt area and a high salt area. So the water will move from the area of dilute salt, low salt, to the area of high salt, right? And so the osmotic pressure is the pressure which is necessary to stop this, to resist this movement. So let's look at that. So here's our pure water. So this is our some kind of solute, right? Dissolved in the water in this test tube. And there's a semi-permeable membrane. So what this membrane does, it will allow water to go through, but not the solute. And because the solute concentration is much higher in the test tube, then water will come from the beaker, go through the semi-permeable membrane, and move into the test tube to the area of low solute concentration. And in a way, kind of diluted, right? Diluted. And to resist this, so let's say there's a piston here, you put pressure on it, push that water back out, and this pressure, which is the force which is necessary for this to happen, is known as osmotic pressure. So get it out of there. So why is this important? Well, because biological membranes are semi-permeable membranes. So here's a cell, right? So it's a cell, there's a biological membrane on the outside. Here we have intracellular solutes, extracellular solutes. So let's say this is a bacteria, right? This is a bacterial cell which want to survive in different environments. And so you put this cell in what's known as a hypertonic solution. So hypertonic solution is a solution of high salt concentration, right? High salt concentration, and the water will move out. The water will move out. And what that will do, that will make the cell shrink. Can you think of an example of this process at home? Something that maybe you have something in the refrigerator that has shrunk because of this placement of something in hypertonic solution?
Fruit.
Fruit? What kind of fruit?
I mean, like any fruit, when it dries out, it'll like shrivel up and shrink.
Yeah. So grapes, for example, right? Like turn into raisins.
Yeah.
Yeah, that's one example. How about something that is a bit salty in a jar?
Pickles?
Pickles. Good example, right? So you have nice fresh cucumbers, put them in a high concentration of salt and vinegar, and that's exactly what will happen to the pickle, to the cucumber, right? It'll turn into a pickle. Now, this is the opposite process. If all of a sudden this bacterial cell ends up in a hypotonic, so this is hyper, right? Hyper, and this is hypotonic. If a cell ends up in a hypotonic solution, then it will swell. It will swell, and the danger is that it may eventually burst, because the amount of this biological membrane is just simply not enough to hold this content together, right? It will stretch and eventually burst. So for that reason, bacteria, for example, have cell walls, right? So we talked about that a little bit earlier. So our cells don't have cell walls, and in our bodies, these osmotic pressure has to be regulated in many other different ways. So very important for our cells to survive, to be able to regulate the osmotic pressure. All right, so let's now switch a little bit to more sort of mathematical description of things. Something that is in biochemistry is very important as well. And all this was in general chemistry. So I will go through this pretty reasonably quickly. So water will dissociate, right, into protons and hydroxide ions. And the equilibrium largely favors water, right? So water does not really want to dissociate. What's the reason for that? I mean, you have a nice neutral molecule of water, stable. Why does it want to break up into proton and hydroxides? So it does it to a very small extent, right? And equilibrium is strongly shifted to the left. And so also keep in mind that once the protons are formed, they combine with water, to form what's known as hydronium ion, right? So the protons don't live by themselves in water. They live as hydronium ions. In fact, hydronium ion is also an oversimplification. The way this actually happens in biological systems is that these protons kind of hop in a solution of water. So for example, if this is the hydronium ion will associate with another molecule of water. So the proton will hop to this oxygen, and then in this molecule of water, this oxygen will reach out and grab this proton. This oxygen will grab this proton, and so on and so forth, right? So there will be a large system of communication. So the protons set up kind of this proton hopping system in place, where they're all over the place. They jump from one molecule of water to another. And at one given moment, you never know what it is. In fact, it's all one big system.
What's the benefit of that?
Well, the benefit is benefit. Well, just think of charges by themselves. So charges are no good, right? So any kind of chemical biological system wants to avoid charges, right? Everything wants neutrality. Charges increase energy. And so by making this proton kind of be associated with not here, not there, so the charge is in a way kind of passed on through this entire system.
Okay.
Right. So even though, and this is probably incorrect, this is an oversimplification. So this charge, because the proton moves away from here, so this charge is diminished, right? So but charge appears, here's a little bit of the charge here, delta plus on this oxygen, delta plus on this oxygen, and so on and so forth. So the charges are distributed throughout the entire system more evenly. And I guess maybe that's the major benefit, right?
Okay.
All right, so a bit more of math. All right, so equilibrium constant. Now remember, this is the product of the multiplication of the products over the concentration of the starting material, water, and can be determined experimentally. Now water, remember, there's in 1 liter of water, we have 55.5 moles of it. And so obviously the concentration of pure water is 55.5 molar. If you plug this all in, you get this number. This is what's known as ionic product of water. And obviously because the concentrations of protons and hydroxides are the same, right? Each molecule of water will give equal numbers of protons and hydroxides. We can split 10 to the negative 14th into the two of each of these 2 to the negative 7th, right? And take a negative log of that. And what we have is that the sum of pH and pOH, now remember this pH, right? So which is the measure of acidity. And so the sum of these equals 14. So 14 is the acidity scale that we use in aqueous solutions. So in a neutral solution, pH equals pOH, and they both equal 7, right? Both equal 7, and the sum of that equals 14. And so here's how they are related, right? So this is, remember, pH is a negative log of the concentration of protons. So if the concentration of protons is 10 to the 0, the pH is, sorry, if the concentration of protons is 1 molar, then pH is 0, right? And then as you decrease the concentration of protons by 1 order of magnitude, pH increases by 1 unit. So the difference, if, for example, the difference between pH 1 and pH 2 is 1 order of magnitude in the concentration of the protons, right? So instead of 10 to the negative 1, it's going to be 10 to the negative 2. So keep that in mind. So pH, the difference in pH, if you have, let's say, 2 units of difference in pH, that's 100 times. 3 units difference in pH, that's 1,000 times. So these are logarithmic numbers. And we don't really use these ones because, you know, once you use pH, this is going to be just the opposite. If this is 0, this is 14, 0, 14. If this is 1, this is 13. This is 2, this is 12. So the sum of these will be 14 in each case. And so we only really need pH. That's why by convention, we just work with the pH and we do not work with pOH. Because one
So
is
just to clarify, because I think I understand what you're saying, is that so the pH are all logarithmic numbers, meaning that they are, like you said, concentrations. So like 7 is 7 times greater than 10 to the 0. Is that what you're trying to say? Or
pHs are logarithms of the concentration of protons. Yes. And so what's a bit counterintuitive is actually the smaller the pH, the more acidic the solution is, right? So 0 will be the highly acidic solution, right? And the reason for that is because it's a negative log. It's a negative log, right? So if this number is higher, then a negative log makes the pH smaller, and vice versa. But because it's a log, it's in orders of magnitude, right? So 1 unit of pH is 1 order of magnitude of the concentration of protons. Is that okay?
okay, thank you.
Yeah. All right, so these are, you can look at these. These are some of the pHs of some common liquids we find at home. You can see coffee is quite acidic, right? So if you have some issues with stomach acidity, keep in mind that drinking coffee will make it worse. And beer too, right? Beer too. Coke, and now things like baking soda, like carbonates, like Tums, right? So when you have high acidity, you take Tums. Why do you take Tums? Because Tums are carbonates, calcium carbonate. So you will decrease the acidity because you add something basic, pH 9. Remember, everything above pH 7 is basic. Everything below pH 7 is acidic. Okay, and remember, very similar equations exist for acids. So this will be the association constant. Well, sorry, dissociation. Well, the opposite. So in this case, this will be dissociation constant. I apologize. So dissociation constant for acetic acid, right? So here's acetic acid interacting with water, where water picks up the proton from acetic acid to give you acetate and hydronium ion. And the dissociation constant is this. And this is the number which has been experimentally determined, right? And so how do you work with these numbers? So this is the dissociation constant. So let's work through one example. So let's say we have acetic acid, right? So we're going to add this to water, and we're going to add 0.1 moles of acetic acid, and add to water, and then fill it up with water to the final volume of 1 liter. So what's the pH of this solution? All right. So before we add the obviously acetic acid, pH is 7, right? pH is 7, it's neutral. Now we add acetic acid, 0.1 molar, right? So a certain amount of it, which is the number of protons and number of acetates, will dissociate, right? So this will break down, and give the equal number of acetate ions and equal number of protons. And what's remaining here is 0.1 minus X, right? So X has dissociated. And so what's remaining is 0.1 minus X. And now there is a new acetate, X moles, and there's a new number of protons, moles of protons, which is X. So dissociation constant is multiply the products, X times X, over the concentration of the starting material, which is 0.1 minus X. And this is this dissociation constant. Now we have a quadratic equation. We'll solve it. We'll find the concentration of protons, and we'll take the negative log of this number, right? Take the negative log, and pH is 2.8. So just roughly, does this make sense? Yes, it does, because pH is now below 7, right? And remember, for weak acids, it's a good number. Weak acids, pH is about between 1 and 6, right? Under 7, but above 0. So we're just in the right region. So it makes sense. So this is one example of a calculation of a pH. Now, there is a simplified method in which we don't have to solve a quadratic equation. And in this case, what we have to keep in mind is that acetic acid dissociates only weakly. It's a weak acid. So which means we can neglect this X, right? So this X can be taken out of the equation because its effect on 0.1 is negligible. So we can leave 0.1 as it is without removing X from it. We still have X and X, XX over 0.1. There's no quadratic equation, very easy. And pH is 2.88. In the previous case, it was 2.883, right? So depending on how accurate we want this to be, we can employ this kind of simplification words. And remember, so pKa is the measure of acidity, right? So for acetic acid, it's 4.76. So it's a negative log of Ka, just like with pH. And the concentration of protons, we have pKa and the dissociation constant of the acids. So carbonic acid here, now we have glycine. So all these are weak acids. And remember, for weak acids, the pKa's will be between 1 and 6. Now, ammonia, amino groups, these can be protonated, right? And so once it's protonated, it becomes an acid because it can lose a proton. It can lose a proton, but it holds on to the proton more tightly. So it's much less acidic, and the pKa is above 7. Okay. Some stuff from general chemistry. If this is still, guys, not clear, you my best advice would be for you to go over this material in general chemistry or even organic chemistry. Organic chemistry, you've taken both. You've seen this both in general chemistry and in organic chemistry. So if you still struggle with this, please look this up, okay? Or put this in a discussion on Canvas, so we can still work through this together. So one of the primary reasons why we need all this is to employ these things in as buffers, right? So what are the buffers? Buffers, remember, are solutions that will resist a change in pH. Whenever you work with an enzyme or a protein, right, they adopt three-dimensional structure. They adopt three-dimensional structure, which is dependent on the pH, right? So in, let's say, highly acidic solution, a three-dimensional structure may be different from in a basic solution, right? So if you want your protein or enzyme to work, you want to keep this three-dimensional structure intact. And so you placed your enzyme in a buffer. You place your enzyme in a buffer of a particular pH. And so the buffering capacity is greatest when the pKa of your acid, okay, the acid now, so buffer is the mixture of the acid and the conjugate base, right? And the buffering capacity is greatest when the pH that you desire equals the pKa of this acid. We'll see in a moment why that is. So also it's important to keep in mind that if you have your acid of a certain pKa, and if you put this in a solution of a pH which equals that pKa, then there will be 50-50 mixture of acid and its conjugate base. So for example, if you have acetic acid, right, which dissociates into acetate and proton, then you will reach the 50-50 mixture of this if you place this in the solution in which the pKa equals the pH. Okay, let's look at this. Let's look at this. So the picture is better than just keep going over this without looking at something more specific. All right, so here's our acetic acid. Here's our acetic acid, and this is the acetate ion. So this is the conjugate base, right? This is starting acid, this is the conjugate base. And so we start with the pH here. All we have is just acetic acid, nothing else, right? We start at a very low pH of about, let's say pH of about 2, starting here somewhere. And we start adding hydroxide ions. We start adding base. As you add base, you start deprotonating acid and moving this into acetate. And as you start adding base, let's say you've added 0.1 equivalents of the base, the pH will rise because the solution will become more basic. And you keep adding, the solution becomes more basic, more basic, more basic. The more base you add, the pH goes up until a certain point after you have added half of the equivalents of the hydroxide ions. So in other words, you have converted half of the acetic acid molecules into acetates. Then you reach the pH of 4.76. You reach the pH of 4.76. So this is what I was telling you. And at this pH, it equals the pKa. This is what I was telling you, right? So when you have half of this and half of that, in other words, half of your acid has been deprotonated in its conjugate base, if you have 1-to-1 mixture, then the pKa and the pH will be identical, and it's 4.76 for acetic acid. And then you keep adding more base. You just keep going. The pH keeps going up until you convert all acetic acid into acetate. But you can see this region here between the pH is 3.76 and 5.76. You can see how much base you've added. You've added between 0.1 and 0.9. So in other words, you've added 0.8 equivalents of the base, huge amount of base, 80%, right? But pH has changed only by 2 units of 2 units, right? The pH has changed by 2 units. And so this is what's known as a buffering region. So acetic acid, so the solution of acetic acid in water is a great buffer, and you can see why that is, right? It can hide the effect of the base so so well, right? So there's so much base you have added, but pH has changed so little. Okay, so this is the principle behind the behind how buffers work, guys. And in the rest of this class, we'll use buffers quite a lot. So it's important for us to understand at this point the basics of them. It's important for us to understand how they work. Are there any questions? Ask me questions.
Would this be the same if you would have added, if you would have added acid to base? Like, would the graph look a little bit different? Like maybe like inverted, I think that's the right word. Does that make sense?
If you add acid to base, you'll go the opposite way, right? So you start here somewhere in the region of high pH, right? If you start adding acid, you start moving in the opposite direction. So the pH will start dropping, and obviously instead of the excess of OH will turn into excess of H plus. So you move in the opposite direction, but you'll still be in the same region. So it doesn't matter which way you go, right? You can add base or you can add acid, but you will still be in between of these two pH units, between 3.76 and 5.76. Yeah, good question. So buffers work both ways, so it doesn't matter. So they resist both base and acid for that reason. Yes.
Okay, thank you.
I have a question. Can you hear me?
Yes. Mm-hmm.
Okay. Does the presence of like sodium ions affect the buffering? Like if you were to make a solution with sodium hydroxide to buffer, would the presence of the sodium ions make a difference in the buffering capacity?
You know, it probably does. I cannot answer your question 100% now because it probably does. I don't know to what extent, but to a very small extent. The reason for why it's why very small extent is because now so for example, you have sodium hydroxide or potassium hydroxide. It doesn't matter because they fully dissociate, right? So when you put KOH or NaOH in water, OH minus is all by itself, right? So there's so the counter ion is not part of the reaction equation. It's a spectator ion. Remember spectator ion from general chemistry? So so it's not part of the equation, but it's still even though we learn to neglect the spectator ions, that's not the that's an approximation, that's a simplification, right, which we use. But if you do work with buffers and highly precise calculations are required, if your enzyme is particularly sensitive to changes in pH, and you really want to make sure that the buffer will work, you will probably use the right counter ion, which has been prescribed in the procedure. Let me read about it myself, and I will post something in a discussion on Canvas regarding that. That sounds good?
Okay, thank you.
Good question, thank you. Anything else? All right, we have a few more minutes. Let's see where we are and what else we need to cover. I'm sure we can keep in connotations. We're almost there. Okay, let's go. Right, so now these are different acids. So acetic acid and acetate, just one type of buffer. So it's acid known as acetate buffer. Now there's a phosphate buffer. You may have heard of that. Also, you can see here, now phosphoric acid has actually 3 protons. So there are actually a number of buffers you can create with phosphoric acid, and the pH difference will be different. There's ammonium buffer. So you can see here, so acetate works in this region of pH, phosphate works in this region of pH, ammonium buffer works in this region between 8 and 10, right? So depending on your ideal pH for your enzyme, you will use the right buffer, right? So there is a reason for, there's a need for a large number of buffers for optimal performance of your biological macromolecule. And some little more math, just to remind you about this Henderson-Hasselbalch equation. This is the derivation of it. Here's our weak acid. We dissociate that, dissociation constant. We pull out the concentration of the protons and take a negative log of that of each of these components. And we end up with this equation. And so what this equation will do for us, it will allow us to calculate pH of a solution of a weak acid, right? Depending on the pKa of this weak acid. And I think there are a couple problems in your sapling homework on the use of this equation. So here is one example. So let's say we have a buffer, 0.1 acetic acid, pKa 476, and 0.05 sodium acetate. What is the final pH of this buffer, right? So we, into this equation, we'll put pKa, right? So pKa, this thing. Where's my pen? Lost my pen. So here's the pKa. We plug this in, it's 4.76. Now going back, oops. The concentration of the conjugate base, and the conjugate base is acetate, right? It's 0.05 molar. And then the concentration of the weak acid is 0.1 molar. We plug all this in, and the final buffer is 4.45, right? So this is how you calculate the pH of your buffer. All right, and of course, buffers, they're very useful for us to work with enzymes, but we're not the first ones to come up with that. Nature has been using buffers for a very long time. And the reason for that is because for each enzyme, there's an optimal pH. Optimal pH, and even things like equilibrium between carbon dioxide gas and dissolved carbonate, right? So, which is crucial for our gas exchange in our system, right? So we inhale oxygen and we exhale carbon dioxide. And so when we inhale, so carbon dioxide in the blood is present as carbonate. When it gets into the lungs, it has to turn into carbon dioxide so we can exhale it. And that will depend on the pH, right? So the pH, for example, in the areas where carbon dioxide has to be exhaled, has to be highly optimized for this conversion of carbonate into carbon dioxide. which is catalyzed by the enzyme carbonic anhydrase. So carbonic anhydrase has an optimal pH it works at. All right, so so here in vivo systems. So in vivo, remember, in vivo means in living systems, in living, right? In living. In living systems, so phosphate buffer, bicarbonate buffer, and we also learn about histidine buffer, which is one of the amino acids, one of the 20 amino acids we're going to talk about in the next chapter. And so there's some buffers based on these systems. Something that if you want more information, you can look it up, I'm sure, on Wikipedia, how these are used in research. And the final, finally we have just a few minutes. So finally, water is also not a solvent, not only a solvent, but it's also bound to proteins and is essential for their function, right? So you can see here in red, I think that's all molecules of water. So this is what I told you about hydration sphere of a protein, but some molecules of water get inside of the protein, right? So you can see here, so for example, yeah, I have something even drawn here. Like there is a hydrogen bond here between this NH, right? And this oxygen. It's too far. If you think about it, the distance from this hydrogen to this oxygen is too large to form a hydrogen bond. But if you stick a molecule of water in between so that this hydrogen interacts with this oxygen and this oxygen interacts with this hydrogen, so there's a very nice bridge. So this molecule of water forms a nice bridge to form a hydrogen bond that otherwise would not be able to exist, right? So this is one of the functions of water, for example, inside of the proteins, is to form a multiple hydrogen bond networking within the inside of the proteins to basically to serve as bridges. So here's another example, right? So here's a protein binds a carbohydrate, and you can see there is water also embedded in the protein in various places. And so these are amino acids. So we'll learn about these 3-letter abbreviations of amino acids in the next chapter. All right, short summary. So we learned about nature of intermolecular forces, properties, structure of liquid water, behavior of various solutes in water, and how water can participate in biochemical reactions. So before we finish, any questions about the chapter on the water? Anything else not clear about water? Wonderful molecule, the medium of life, and you can see there is a reason for that, right? Lots of properties, lots of possibilities for biological molecules through their contact and various forces that develop between them and water. Okay, so I will place this on YouTube. Now, homework, don't forget, guys, the homework is due, when is it? Wednesday, right? Homework is due on Wednesday. I would like to encourage I would like to see more activity in the discussion section on Canvas. That's how we learn. And since we don't have a class environment, and of course you can always email me. And I also have office hours which follow this lecture. All right. Have a good week, and I'll see you on Wednesday.
You too. Thank you.
You too. Thanks.
Thank you.
Dr. Kornicko, will you be posting the chapter 3 PowerPoint today or sometime this week?
Yeah, before we start, definitely.
So before Wednesday, just so I can print it out.
Before Wednesday, yeah.
Okay, thank you.
Mhm.